My name is Amanda Pritzlaff, and I'm a graduate student in Dan Savin's lab at the University of Florida. Today, I'm going to discuss my research with you and what makes it so interesting. I work on generating something called protein-polymer conjugates. Let's understand what this is by looking at each component individually. Proteins. When you think of proteins, you probably think of something like this that you consume to help make you stronger. However, proteins are not just an essential nutrient, but fascinating molecules with unique functions. Your cells couldn't keep you alive without the thousands of proteins functioning within them at every moment. How do proteins carry out these complex functions? Proteins are giant compared to other small molecules and also structurally complex. So let's get an idea of molecular size. Here you are, and let's compare you to a small molecule like aspirin. The size difference between you and aspirin is 10 orders of magnitude. To understand that size difference, let's take a grain of rice. Compare the size of a grain of rice to the Earth, and that is the same size difference between you and a molecule, 10 orders of magnitude. Proteins are giant molecules. This size comparison would instead use many grains of rice connected in a line and folded in a defined 3D shape. Now, instead of rice, each unit of a real protein could be one of 20 different building blocks, shown here. This causes proteins to have diverse sequences like the one on the left and fold into a defined shape, color-coded on the right. The protein I work with is called galectin-3. Its structure is shown on the left. It is produced in humans, and its structure allows it to bind sugars or carbohydrates, shown on the right. These are another important biological molecule. We hope to harness this protein's sugar-binding function and incorporate the protein into materials that will bind sugars. This is where the power of polymers comes into play. Part 2, polymers. When you think of polymers, you probably think of plastics that have amazing properties like hardness and impact resistance. Polymers have these properties because they are giant molecules, just like proteins. However, polymers are long, disordered molecules that do not fold into ordered shapes like proteins. Instead, the chains tangle together like a string of beads and pack tightly. Each chain interacts with neighboring chains and moves slowly. Here we have a structure of a common polymer with some repeat units called monomers circled. Protein monomers are circled as well to show that a protein is really just a complex polymer. In my work, I attach or conjugate galectin-3 to polymers and see if this changes the protein's ability to bind sugars. Then the protein can be attached to a polymer support and used to purify sugars from mixtures. This can be used to optimize industrial processes.